This is a production of Cornell University.
Thank you, Evan, for that introduction. As you said, I'm Dustin Wilkerson. I'm a third-year PhD with Larry Smart. So I moved to Geneva about a year ago, and so that's why some of you may not even recognize me. I've been up in Geneva. So hopefully by the end of this talk, you'll have some idea of what I've been up to. I have actually been doing things up there. But I'm going to talk about one component of my research, which is using this common parent F1 hybrid mapping population basically to find resistance to willow leaf rust. So I know Brennan gave a talk last week on shrub willow, but I wanted to give sort of an update. So shrub willow is a very diverse, or Salix is a very diverse genus. There's roughly 300 species. They come in tree and shrub form, a lot of different ploidy ranges, and a lot of them hybridize within each other. So there's a lot of diversity to play with, and it's really interesting sometimes. It has a variety of uses between biomass grown for bioenergy, phytoremediation, because it can grow on soils that are typically unsuitable for other crops. It's very hardy. And also just as physical barriers, like privacy fences or alongside roadways as like snow blocks. It is dioecious and highly heterozygous, so we have male and female plants, which makes breeding interesting. And willow that is grown for biomass is grown in a short rotation coppice system. So this means we plant cuttings, they're cut back after 1 year, and then they are harvested for chips every 2 to 3 years afterwards. So these plants can stay in the field for 20 plus years before they're switched out. So you can imagine kind of in this picture here, it's a very dense kind of planting structure. And so diseases and insects can really thrive and take hold in these kind of situations. And so the most damaging of them being willow leaf rust, a relative of the talk that we just heard from Shatai. So it can lead to complete defoliation in a year, and it can result up to a 50% total yield reduction. It is also macrocyclic and heteroecious, so this means that we'll go through all 5 of the spore stages that we heard about before, and requires an alternate host to complete them. So the alternate host for the figure I have here would be the balsam fir, so it's more of a tree rust. Like I said, it's got that potential for sexual recombination each year. So you can imagine when you've got willow that's in the field for 20 plus years that may have been resistant to rust when you planted it, you have something that's sexually recombining every year. So that resistance is probably not going to be there after a few years of sexual recombination. And it is described mainly by these yellow to orange pustules that are on the underside of the leaf. And this is how we use to phenotype severity based on the percent coverage of a leaf. Using these spores. So because it is so complicated, there's a lot of diversity within Melampsora. And like I said, there's a lot of diversity within Salix. We wanted to try and kind of use that diversity in Salix to identify resistance genes that can be sort of pyramided into mixture plantings in that SRC system. So typically they're grown in monoculture, but the best form of management, the most effective, is to mixture plant with different levels of resistance. So obviously we need to know about those resistances before we can advise what kind of mixtures need to be planted. And so 2 different mapping populations were constructed to get more information on shrub willow. So the first one is a Salix purpurea F2 population that kind of takes center stage in other aspects of my research, but for the time being, I'm going to talk about the other 8 families that are kind of surrounding it. So these 8 families comprise the common parent, what we call for short just the common parent mapping population. So we utilize the naturalized Salix purpurea, which is the 946 and 9401 as these purple boxes. So 4 families share. It's my instinct to move with my hand, but I'm going to do this now. So 4 of these families share 9406 as the common female parent, and the other 4 will use 9401 as the common male parent. With Salix suchuensis and viminalis having a reciprocal cross within it. Salix viminalis is important because it's the most widely grown species of Salix in biomass in Europe. And so a lot of the studies in this system have been conducted using Salix viminalis on European species of Melampsora. So all of these individuals have been sequenced using GBS. And so I put together this PCA of those 8 families. So you can kind of see the common parents, 9401 and 9406, are kind of splitting the families, where I'm doing it again, see? 9401 and 9406 are kind of splitting the 2 families up, where when we look at PC1, we can see that these 2 families here are the Salix viminalis that I just mentioned, where this one is Salix udensis. So PC1 is really separating these families phylogenetically. So these 3 are in the section Vemen, where the families on the other side of the PCA are from the section Helix. So they're more related. Where PC2 is doing the work of separating out each family. So you can see we have this family here with the F1 kind of lying somewhere in the middle, as we would expect it to do. So now that we have our GBS data, if I want to map QTL, I need to construct maps for everything. So I put together this general pipeline of the way that I handled that GBS data. And so I used Tassel GBS to call variants, which, depending on the family, resulted between 3 and 500,000 markers in each family. These were then passed into the Tassel GUI, and I filtered for up to 80% presence, which really cut down those number of markers. They were then subset in R for backcross markers. So it then, instead of dealing with one population, now we're dealing with 2 separate maps for each one, for each parent, based on the parental genotype. And then I used a combination of RQTL and ASMap to do some additional filtering and for determining the linkage groups and establishing the marker order. And so each map at the end of this process has between 1 and 3,000 markers. So we have 16 maps in total, 2 for each of the families. So again, now that we've got the maps, which makes it sound so easy, now they've got the maps, we need the phenotype data to really kind of bring it together. Unfortunately, leaf rust has not been very cooperative with me in this study. So in the years 2016 and 2018, we had negligible to no leaf rust presence in the field at all. So we're really hoping 2019 we can actually get a good survey. But the one that we were able to do, 2017, in reality is a mild rust year. But we can still, there's still things to learn from this. And I should mention that all of these families are planted up at Cornell Agritech in Geneva. There's 4 replications. It's about 4,000 plots for all families together. And so they've all been surveyed. So now that I've had this plot up for a little while, let me try and explain it a little bit. So the 2 purple bars are the common parents. So there's 9406 here and 9401. The vertical line in the middle kind of delineates the families that are using that common female or common male parents. So as you look at each F1, which are these larger boxes, the female is always on the left of the F1 with the male next to it. So you got the female, male, F1 male, and then it switches on the other side. Each dot represents a genotypic mean to kind of give you an idea of, even though the mean is here, we still have a wide range of leaf rust severity through these populations. So when I finally got this figure worked out, one of the first things that jumped out to me is how each family kind of has its own story with leaf rust. So in the case of this family here, the 407 family, we can see that the mean of the F1 is kind of between the parents, which is what we think more of as an additive model with between the 2 parents. But if we look at 443 on the end, there seems to be a breakdown in resistance. And that's common throughout some of the other families as well. So even though, like I said, even though this was a mild year, we're still getting story about leaf rust with each of these separate families. And so it's important to kind of think of them as a population, but also as an individual families as we kind of think about the results that we find. So now that I've got the maps constructed and the phenotype data, I actually was able to identify a few QTL. And so I just pulled one out so I could show you. So this is a map from one of the Suchuensis families. It's a map of the female common parent that I put together. And we were able to find a QTL on chromosome 7. So you can see this kind of national monument-shaped QTL here. Yeah, it has this nice peak here, which actually, maybe a little bit lucky, is a marker. It's an actual marker. And so pulling that out and looking at the allelic effects, you can see that anything homozygous at that marker, there is still a rating here, but a lot of them are at 0, and it's very mild, whereas anything heterozygous at that loci has a higher phenotype, suggesting maybe it's conferring maybe partial resistance at that loci. So going forward with these maps, I really want to, as I've said a few times, each family kind of has its own personality. And so I want to go through each of the genetic maps and kind of address some of the eccentricities there before I finalize them and am content with their status. Once that's done, I'll be able to go through and finalize the list of leaf rust QTL that I was able to find. And naturally, I've been going through those fields for a lot of separate traits. And so I want to use those traits as well. And I would also like to try a way to analyze all 8 families together. I'm kind of bouncing around with that, but I want to make sure I get these maps done before I move on. So with that in mind, I'd like to thank everyone for coming. And I'd also like to thank everyone at the Larry Smart Lab, with a special shout out to Rebecca, Lauren, Jane, and Dawn, who are our technicians. Pretty much everyone here knows the lab would fall apart without them. So we're extremely grateful for them. And also Holly and Chase from the Chris Smart Lab. So I'd like to thank everyone again, and I can take questions.
I have 2 questions. One, in the years when you don't have rust, or the time you don't have rust, could you somehow artificially inoculate some of the plants to look at the differences in phenotypes?
So the question was, in years that we don't have rust, can we artificially inoculate some of the, is that right? Yeah.
Yeah.
So I know that has been done. So I mentioned Chase Crowell. He's kind of my partner in crime in plant pathology in Chris Smart's lab. So he has done some work with different isolates of rust in these different families. But as far as what my work is done, it's really important that we can get field-based measurements. So,
okay. To follow up on that, the second part of the question is: are you looking at complete resistance, or just tolerance? Cumulative tolerance to stone rust be good enough for what you're using the Willow for?
So the question was, are we after complete resistance, or would some partial resistance be enough? And honestly, as I said, when you're dealing with a mixture planting, it's really best to have different levels of resistance, because obviously you don't want to encourage kind of that selection. You kind of want to you almost want to let it have something so that there isn't a lot of selection pressure. So in terms of if we care about absolute or partial, I think I'm open to whatever I can find. As long, it's really about kind of characterizing what we have here.
Thanks. I have a couple of other quick questions.
So in Chai's talk, you talked about all these different races of the rust. Is it known what the race structure of the rusts are for this Willow?
So the question is, what if we know about the race structure within Melampsora? So there is a lot of work on that. I know Chris Smart's lab has done some work on that, but definitely compared to wheat rust, we don't know very much about this. So a lot of what we've been able to find is those different species of willow obviously respond differently to the different races. So we know that if we can go into a field in any given year, we're probably going to find different races within a single field. But I don't know if it's really been characterized to them.
Please join me thanking Dustin one more time.
This has been a production of Cornell University, on the web at cornell.edu.